How do targeted therapies specifically fight lymphoma? Imagine fighting lymphoma with a precision tool that targets only the cancer cells without harming the healthy ones. That's exactly what targeted therapies aim to do. Instead of traditional chemotherapy, which attacks all rapidly dividing cells, targeted therapies focus on specific molecules involved in the growth and survival of lymphoma cells. This makes treatment more precise and often reduces side effects. One way targeted therapies work is by blocking signals that tell lymphoma cells to grow and divide. For example, some drugs inhibit proteins like Bruton's tyrosine kinase, which is essential for B-cell lymphoma survival. By blocking this protein, the therapy can stop the lymphoma from growing or even cause the cancer cells to die. Similarly, drugs that inhibit phosphoinositide 3 kinase, another key protein in cell growth pathways, can slow or halt lymphoma progression. Another approach involves using special antibodies that recognize proteins on the surface of lymphoma cells. Rituximab is a well-known example that binds to CD20 proteins on B-cell lymphomas. Once attached, the immune system can recognize and destroy these marked cancer cells. Brentuximab vedotin targets CD30, a protein found on certain lymphoma cells, and delivers a toxic payload directly to the cancer, sparing most normal cells. Targeted therapies also help the immune system fight lymphoma by blocking proteins that cancer cells use to hide from immune attack. Checkpoint inhibitors like nivolumab and pembrolizumab block PED1 or PED-L1 proteins, which cancer cells exploit to evade immune detection. When these checkpoints are blocked, the immune system can better recognize and attack the lymphoma. Cellular therapies such as CAR T-cell therapy take this a step further. In this treatment, a patient's own T cells are collected and genetically modified to recognize lymphoma-specific proteins. These engineered T cells are then infused back into the patient, where they seek out and destroy lymphoma cells with high precision. Some targeted drugs also interfere with the internal machinery of cancer cells. Proteasome inhibitors disrupt the breakdown of proteins inside the cell, which can lead to cancer cell death. Others, like tazemetostat, target epigenetic regulators that control gene expression, affecting how lymphoma cells grow and survive. Choosing the right targeted therapy depends on the specific type of lymphoma, the proteins expressed by the cancer cells, and genetic mutations present. Because each lymphoma is unique, treatment is often personalized based on molecular testing. While targeted therapies have improved outcomes and are often better tolerated than traditional chemotherapy, lymphoma cells can sometimes develop resistance. They may do this by changing the proteins they express or by finding new ways to evade the immune system. That's why ongoing research focuses on combining different targeted agents and developing new ones to stay ahead of resistance. In summary, targeted therapies fight lymphoma by disrupting the molecular pathways and proteins that cancer cells rely on. They can block growth signals, mark cells for immune destruction, deliver toxins directly to cancer, or modify immune checkpoints. These approaches are helping to make treatments more precise, effective, and easier to tolerate. Always remember, treatment choices should be discussed with healthcare professionals who can tailor options to each individual's unique situation.